Hi everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. Although today is not going to be a lecture, it's going to be a laboratory presentation. The college opened up for a short period of time, for one day, for some of us to come up and get some things done right before registration. So I'm here doing this. We're in the coronavirus shutdown. These are going to be quick, impromptu videos done with my cell phone. So you'll see me leaning over and other things. What I'm going to do in this series of lab videos is I'm going to make short videos covering each model. And what I'm going to do is I'm going to draw it out on the board because I do believe that you understand it better when you see it unfold. And then, as unprofessional as it may look, I'm going to come over, grab the camera, set it up so that we can go over the model together. All right? So I'm going to try to do all my lab stuff in one day, a bunch of quick videos. So please bear with me. Also, I have to have the lights out or else there's glare all over the board. I hope you can see. Okay, so now, what we're doing is this is going to be lab test number 4. These are the labs for the nervous system in 2401 A&P, or Part 1 A&P. So we're going to do the neuron model and we're going to do the synaptic knob model, and we're going to understand them. This will also help us for lecture with the anatomy. As you guys know from lecture, neurons come in different shapes. There's anaxonic neurons, there's unipolar neurons, bipolar neurons, and multipolar neurons that we've looked at. We're going to be focusing in on a multipolar neuron because that's what our model shows. And I'll just give you a quick look at what the model looks like. Pardon my big belly, but we're going to be looking at this model. We're going to look at it like this, okay? So hopefully you can see that. I can't even see what I'm looking at because of the way my camera is set up. So anyway, bear with me. So when we look at a multipolar neuron, we know that a multipolar neuron looks like this. It'll have lots of branches off of this soma, and then a single branch going in this direction that would branch out like this. These would be dendrites, and they would be hugely branched. I mean, if you look up pictures of these things online, it's amazing how large and how branched the dendritic tree can look like. It's just stunning. Our model cuts all of the dendrites off like this, and we focus on a couple of dendrites, the soma, and the rest of the neuron. But they also cut the neuron like this, so we don't even see the synaptic knob or the telodendria. So I'm going to go over the anatomy of this model with you really quickly. So the model should look something like this. You'll have these little white branches sticking out like this, and these are the cut dendrites, okay? There'll be a long branch that comes down like this. And even though they don't show them, I am going to put a couple of telodendria and synaptic knobs here, or a few of them, okay? Now, inside of here would be our nucleus. Surrounding the nucleus in a neuron, there's an area that kind of picks up a darker stain. I'll use this purple color, but there's sort of a darkly staining area here. And what it is it ends up staining a lot of the organelles. Now, the guy who first studied neurons, as I said in the lecture videos, named this the caryon. We didn't know what the nucleus was. So the area that stains very darkly around the nucleus is called the perikaryon. Karyon means kernel or seed. Perikaryon means around it. If I could, and I'm going to, if I made a dotted line here and cut off all of the branches, then this entire central piece would be called the soma. Soma means body, so it's also the neuron cell body. And then every one of these branches would be called dendrites. Now on our model, pardon me, I'm going to be reaching across here. I'm going to try not to, but there are these small structures that are sitting on here like this. I don't even know if that color shows up very well, so let me get a different color. Let's see if this one's any better. And these small structures that are sitting here like this are not dendrites. This is the telodendrion and the synaptic knob of other neurons releasing neurotransmitter onto this one. So telodendrion, synaptic knob, we're not really going to worry about that. Telodendrion, synaptic knob. But I just don't want you to confuse those things. Now, where the axon is, this long branch here, this area, and on our models, it's got sort of a, it's been colored in with orange. This orange marker is not working very well. Let's see if I can get another one. It's been colored in with orange here. And that area is called the axon hillock. And then this entire branch here would be called the axon, going down to the telodendria and the synaptic knob. So it's pretty simple anatomy. Soma with the nucleus and perikaryon, the dendrites, axon, hillock, and then the axon. Now, some neurons are myelinated. Myelin is this fatty substance that has a white appearance. And so it makes up the white matter of our nervous system. Anytime you see white somewhere in the nervous system, it's white matter. If it's myelinated, we call that white matter. Where we lack myelin, the tissue appears sort of a grayish color. So we call that gray matter in the nervous system. And it's usually unmyelinated structures. Now, if you see myelin, it's myelinated axons, if it's white. If you don't, then it can either be unmyelinated axons or it is the somata. Somata is plural for soma. And the dendrites. Dendrites and the somata are never myelinated. Myelin was found in these cells called Schwann cells, named by a guy named Dr. Schwann. So what these cells do is they will wrap around the axon and insulate it. They electrically isolate it or insulate it. So, and they do so kind of like paper towels around a paper towel roll. There's multiple layers of these cells. So I'm going to draw a few of them. I'm going to draw a couple of them kind of covering the axon. And what they would do is they would wrap their membranes around and around and around a whole bunch of times, and they extend a certain length. So if I just cut the end of this one off so we could see it, and then they end, and it would be covering the axon. We wouldn't be able to see the axon underneath here. And then there's a small gap, and then there's another one of these Schwann cells. And then on our model, we're going to do a third one here. There's really many of them along the axon of a neuron. Now, the first person to discover these called it a myelin sheath. A sheath is something rather soft that you would stick a sword or a knife in. If it's hard, you call it a scabbard. And so anytime you take a long sword and you stick it in a soft like leather casing or something that you would clip on a belt, we call that a sheath. And because they discovered that this was a fatty substance called myelin, and it completely encapsulated the axon, then they called all of these Schwann cells, all of these cells covering the axon itself, here I'm going to write axon in here, okay? All of that is called a myelin sheath. Now, later on, with better instruments, better microscopes, someone studied these and a guy realized it's not just one big tube of fat, but it's actual cells wrapped around here. And so each cell that makes up the myelin sheath is called a Schwann cell. Another guy came along and decided, well, I see these little gaps between the Schwann cells. And he's a French guy, so he called them nodes of Ranvier. Some people will say Ranvier, but they're nodes of Ranvier, okay? So the myelin sheath is all of the Schwann cells that cover a single axon. And then if I look at them individually, they're called Schwann cells, and the little gaps between them are called nodes of Ranvier. Finally, there's some real funny lingo here. We call the cell membrane of a typical cell the plasma lemma. We use the lemma, L-E-M-M-A, from lamination, which means layer. On muscle cells, we call the cell membrane the sarcolemma. On neurons, they call the cell membrane the neurolemma. And you'll see this term sometimes with an O. Now, it turns out that, and the cytoplasm inside a neuron can be called neuroplasm. The covering of the axon has some slightly different properties. There's a distribution of ion channels that is somewhat different. So they call the membrane that covers the axon the axolemma. And then they call the membrane of the Schwann cell the neurolemma. Neurolemma is the cell membrane of the neuron. It's N-E-U-R-O-L-E-M-M-A. The neurolemma is the cell membrane of the Schwann cell, N-E-U-R-I-L-E-M-M-A. So that's the neurolemma, the neurolemma, the axolemma. And the axoplasm would be the cytoplasm. So our models have been cut right about here. And when we look on the end, we'll see the end of the axon. We'll see some axoplasm inside there. It's actually colored the same as that up there. It's kind of an orange color inside of here. And then you'll see the Schwann cell wrapped around here multiple times. So I'm going to try to present to you this model so that you guys can see what I'm talking about. Okay, so give me just a second to get set up. Let me get my model ready here. Let me switch on the light. Maybe you can see a little better. And you can follow along here. I'm going to